Now, unlike our TLR receptors, which were going to be membrane bound, we also have what are known as the NOD-like receptors, NLRs. These are going to be nucleotide binding oligomerization domains. Basically, they're intracellular sensors that are floating around in the cytoplasm of the cell. Now, as they're floating around in the cytoplasm of the cell, they are not looking for specific proteins. What they are looking for is specific dipeptides or specific domains or DNA sequences that are not human. Whether it's going to be, things like glutamyl diaminopelic acid, breakdown product of peptidoglycan. So you have an intracellular bacteria that has escaped into the cytoplasm. You'll see this with Salmonella, Listeria. Well, if it's already in the cytoplasm, toll-like receptors on the surface and pointing out into the extracellular matrix have nothing to do anymore. So now you have the NOD receptors. NOD1, NOD2. NOD1, NOD2 will bind to something bacterial. Something bacterial binds to it, dimerizes, kicks off a cascade. Kicks off a signaling pathway that, again, NF-kappa B gets activated. You get the expression of cytokines, interferons. Interferon will tell this cell to amp itself up, and will also tell this cell to begin phagocytosis, begin digesting whatever may be in a phagosome. Also may trigger more autosomal. Time to maybe do some autophagolysosome, start digesting away some stuff. Also will then recruit in other cells to tell this cell to kill itself. We also have what are referred to as caspase recruitment domains. A subfamily of the NODs. These guys recognize caspases. Caspases are are a series of signals that tell cells time to die. Caspase 1, 2, 3. It's a domino effect of proteins that will ultimately trigger the destruction of the genome, the breakdown of the mitochondria, death of the cell through apoptosis. Now, if there happens to be some damage to the cell, whether it's an injury or a pathogen, you get some sort of a pathogen that's now damaging directly the cytoplasm through either an endotoxin, exotoxin, or maybe it's a parasitic worm. NALPs, which you see right here, are another intracellular protein free floating around in the cytoplasm. NALP3 associates with certain proteins, keeps it in an off setting. Keeps it in an off setting until potassium. We keep as much potassium in and much sodium out. Remember the sodium-potassium pump you learned in 244. Well, when there's damage to the cytoplasmic membrane, potassium leaks out. The low potassium environment will cause certain caspases, caspase 1 specifically, as well as picard, to cut themselves. They cut themselves, and then they associate, force NALP3 to dimerize. This dimerization and this cleavage triggers, again, downstream effects, which will cause for the increase of IL-8 and IL-1 beta. I'm sorry, IL-18 and IL-1 beta. These are cytokines to be cleaved and released. IL-1, IL-18 will trigger the attraction of other cells to come tell this cell time to die. But hey, what happens if it's a virus? Everything we've been talking about up to now has been mostly bacterial. What do you do if it's a virus? Well, viral RNA has certain sequences that are going to cause secondary structures, or it's going to be a double-stranded RNA. The secondary sequences, the secondary structures, as well as the double-strandedness are completely foreign to us. So we have proteins. Proteins such as RIG-I. RIG-I binds to triphosphate RNA. This triphosphate RNA is viral origin. It is not going to be mammalian, eukaryotic in origin. So, RIG-1 binds specific RNA sequences via this helicase domain. This RIG-1, retinoic acid inducible gene 1, is going to bind, like I said, viral only. It's not going to interact with our messenger RNA. Because of the secondary tertiary structures of tRNA as well as ribosomal RNA, RIG-1 cannot interact with them. RIG-1 interacts with viral RNA, so we're looking at RNA viruses here. Those bound together will then interact with CARD domain proteins, specifically MAVs. As more of these RIG-1s interact with RNA, they'll interact with the MAVs. You get this dimerization occurring more and more, till you get a certain number interacting together on the surface of a mitochondria, which will then bring in proteins such as TRAF6, FAD, TRAD, which will go through and start their own signal pathways, ending with IRF3. IRFs, remember, these are going to be antiviral signals, and then NF-kappa B, which will traffic to the nucleus, causing for more cytokines as well as interferon to be expressed. What does this do? Well, you can get if you follow it through, bacteria bind to the endothelium, or this could be the mucosal lining of your mouth or wherever. Wound causes break in the defense. Bacterium get down in. Langerhans cell, a type of dendritic cell, will then take up the bacteria. The bacteria has been coated with complement. Or the LPS specifically binds to CD14, or you get interaction from toll-like receptor 4. The Langerhans cell will then take up, digest this bacteria, and in so doing, the Langerhans cell will then migrate, travels up the efferent tube to the nearest lymph node, and then starts going through the different areas until it finds macrophages, tells them to move, and then it looks for CD4 T cells. These are the helper T cells. By activating the helper T cells, it will then go and tell B cells, as well as the CD8 cytotoxic T cells, that it is now time to go to work. Now, we have found that while we know quite a bit about the toll-like receptors in humans, in mammals, it's very hard to go through and to change things up. Since we have so many chromosomes and so many different things going on, genetic manipulation on us is quite difficult. So what we've done instead is we have looked to toll-like receptors in Drosophila, basically the fruit fly. Nobody cares if you screw up a fruit fly. So we've gone through, we've mixed things up just so we can see what happens. What we figured out, bacterial peptidoglycan, the cell wall, binds to two different PAMP receptors on the surface of a cell in the Drosophila, triggers protease cascade, and then a protein known as spatzle will be cleaved. The cleavage of spatzle will result in the dimerization of toll. That's where we got the name toll-like receptors in humans and mice. Toll was the original protein, the original receptor we found, and we found it in fruit flies. The dimerization here causes the dimerization of the cytoplasmic tails, which starts a whole downstream cascade. And then we see some form of NF-kappa B gets released from its I-kappa B inhibitor, and then the expression of different cytokines and chemokines. Now, when it comes to the cytokines, cytokines can either act on the self, the same protein the same cell they came out of, or things in their vicinity or elsewhere in the body. Things that are known as autocrine. Auto is self. So these cytokines are going to be released and immediately have effect right on the cell as it binds that cell's receptors. So this cell gets to turn itself on, gets to amp up its own response. If that cell releases and then it activates cells in the same tissue, the same organ, that's referred to as paracrine. If the cytokines are released and then get into the bloodstream, that's endocrine. One of the different types of cytokines we see a lot are interleukins, IL. These are going to be molecules secreted to act on leukocytes, the white blood cells, to turn them on, amp them up, or to downregulate them. We see that the interleukins, the interferon, ILR, interleukin receptors, the interferon receptors, the tumor necrosis factor receptors are all grouped by classes. So you see here, IL-1 family receptors, they all have that specific domain that we saw back when we were talking about T-cell, B-cell receptors. Here, the same thing. Receptors for GM-CSF and other cytokines, they have a semi-quasi MHC look to them. TNF, and then chemokines and FMLP receptors are this multiple transmembrane looking thing. So again, we go back to our friend the macrophage. Macrophage gets triggered, its receptors gets triggered, signaling pathway gets activated, and then a whole host of cytokines are released. The five biggies: IL-1 beta, TNF alpha, IL-6, CXCL8, and IL-12. They can either have local effects or systemic effects. Systemic effects means it's going to flow through the interstitial fluid, taken up by the blood, and then travel throughout the rest of the body. Recruiting. Depending upon which class of chemokine, it's going to tell you which cells are going to be brought in. They'll be produced by macrophages, monocytes, different cells, and here's who they recruit. The recruitment, it's not just a here we are, come here, but it can also activate. In the process of recruiting, it will also activate specific abilities in the target cell.